here, but I really just got through some of the introductory stuff to that paper or the assigned paper. You are still going to be responsible for this paper, okay? So what I want to do is go through not everything that I did on Friday, but I want to spend a few minutes before we get talking about the next paper on global expression, just to make sure. I think if things you might not understand about this assigned paper with the APOE, I think it will become clear when you watch the video from Friday, and I'm going to highlight a few things here more global, big perspective. So I'm not going to go through all of these, but do pay attention to the things that I've got on this slide, because I've added information that I think will help you understand. So the main thing is that they're differentiating into 3 distinct cell types: astrocytes, microglia cells. I'm not sure why they call them microglia-like cells in the differentiation protocol, but we'll trust them in their neurons. And they have very specific differences with respect to their contributions to Alzheimer's. It's pretty clear there's an interplay between all 3 cell types, and that's been one of the major deficiencies of any of the up until recently, any of the culture methods, the in vitro methods where you culture cells and try to reproduce a disease mechanism. If you've only got a purified population of cells, well, that's not at all recapitulating what's in a tissue. Okay, and there's lots of communication. Even though there's not a primary defect in one cell type, and there is a primary defect in another cell type in that same tissue, there's still that other cell type can be influencing the degree of disease progression, okay, in the affected. So we have to have better models for understanding how the gene products from different cells interact with each other, especially in the case of Alzheimer's, where we know that cholesterol outside of the cell and remember the A beta proteins, those polypeptides that are produced from the amyloidogenic, the pathogenic pathway. We've got to have better modeling for all of those things. Now, I would say this is just a first step towards that, being able to take isogenic cells, right? Everybody know what isogenic is, right? So they got iPSC cells that they did gene editing to change the ApoE allele, but then they are able to differentiate the exact same cell types, ApoE genotype versus ApoE3 genotype versus ApoE4 genotype, differentiate them into 3 separate pathways, 3 separate cells. Okay. Now, and then the last set of experiments are really very interesting, getting at this idea of how are those cells communicating with each other in disease. But first, I mean, I think of this paper as really just kind of being the proof of principle of being able to do this and establishing these differentiated cells and establishing firmly, I think, that there truly are some fundamental differences in how each of these different cell types behave if they've got an ApoE4 homozygous genotype versus an ApoE3 homozygous genotype. It's never been addressed before. So you don't have to know about all these details, I won't test you on them, but just so you know, they're using very well validated methods for differentiation, okay? Now, some of these things they will be kind of referring to, but just for right now, just don't think that ultimately they're getting neurons, they're getting astrocytes, they're getting microglia-like cells. Okay. But they basically did, this is kind of the big picture I want to tell you about if you haven't seen it yet with this paper. They basically did the same set of analyses with each of the 3 different cell types. First, they had to show more convincingly that they truly are neurons, truly are astrocytes, truly are microglia. So they validated their cell type. Then they did expression analysis to see what effect the different ApoE genotypes had on overall expression. Okay, and that's really kind of important to study. So this gets to, and I discussed this in the lecture Friday. Be sure you watch the video. I mean, these are really kind of important, not so much towards the ultimate main conclusions, but the fact that these groupings are important, that the two genotypes of the astrocytes clump together, group together, two genotypes of neurons, two genotypes of the iPSCs before the differentiation. That was important too, because they need to make sure whether the ApoE4, ApoE genotype is having like this global effect on either differentiation of the cells or making them look different than the others. Now, this is really pretty striking, this difference between the microglia cells depending on genotype. And this is reflected in there definitely is a very distinct difference between these cells and these cells, okay? And it has to do with, I think, functionally significant. So this is just, DEG stands for differentially expressed genes. So these are, transcriptome, I think this was RNA-seq, for the three different cell types. Now still, they're not doing anything in terms of looking at specific pathways having to do with ApoE function. They're looking at global gene expression to see if there's any large-scale differences in gene expression. And because these orange ones are the ones that are significantly different. This is consistent with what we saw on the previous slide, is the microglia cells really have some profound difference with respect to genotype. And that's followed up further. And then kind of doing the same thing, just plotting it differently. But now what they're doing is comparing each of the three different differentiated cell types, neurons, astrocytes, and microglia, against genes that are expressed at differential levels in post-mortem human samples, human brain. Okay, now they didn't have Alzheimer's patient brains, but they had mild cognitive impairment. So these could be like, we don't know how many of these people might have gone on to progress to Alzheimer's, but they clearly have some altered cognitive functions that could be related to early symptoms of Alzheimer's. So they were able to pick out a lot of the same genes that are being differentially expressed, strictly on the basis of the differences between the ApoE genotypes. Okay, so that's telling them that they probably have a pretty good cell knowledge, okay, for understanding. If they hadn't done these experiments, I don't think that you could critically evaluate the results that they got when they started looking at specific functions that might relate to cholesterol metabolism and Alzheimer's, progression of Alzheimer's disease. Okay, so I think these experiments are really very important. And this is just a different representation of it. Now they're doing direct, these are not heat maps with respect to expression levels, they're heat maps with respect to concordance, matching values, relative values between the brain samples and their differentiated cell types. Yes? Yes.
Could you just go over like when we integrate, how to integrate each one of these?
Yeah, so with each panel is for a separate differentiated cell type derived from iPSCs. So on the left are the neurons. So what they look at these axes. Now EQTL is a term you might not know, but do you all know what quantitative trait loci are, QTLs? Okay, so they're like GWAS, only it's kind of an older term, and there's just some different technical differences between an association on the basis of a GWAS and a QTL. Does anybody want to add anything to? Do you have a better description of what a QTL is? Anybody? But what this is doing is letting them look at a specific gene, but EQTL refers to the expression level of that gene, whether there are quantitative differences in the expression level of that gene that relate to the phenotype. That was done in the previous studies with respect to mild cognitive impairment. Okay. So on this scale are the EQTL values for these genes, and then on this are for the very same genes cultured neurons. Okay, so then again, the blue dots are all the genes that are not significantly different. You'd expect a 1-to-1 for the vast majority of them. But the ones in this lower left quadrant are those, is the relative expression in the postmortem brain samples higher or lower for these genes than in the called iPSC-derived neurons? Are these higher or lower in the brain versus the neurons? Okay, so you're going here, if you go here to there, that's 1 to 1. Here, it's slightly lower in the neurons and still higher. So I would say higher in the brain. Just the opposite over here. These are being upregulated only in the microglia cells. These are, relatively speaking, these are upregulated in the brain.
And I'm so sorry, I have another question. When you say postmortem brains, like how did you read that from?
This is from a previous study.
Okay.
So yeah, look at it, sorry, that's, I don't have that on the slide, but that's described in the.
down here upregulated.
Okay, now we're
She said
getting
this is
into
hard.
characterizing the cell types. So now they are different cell types: neurons, astrocytes, microglia. So there are different details of how they're establishing that they truly are astrocytes, and that the ApoE genotype isn't having a major effect on them. Okay. So there's different sets of markers or assays that are being used. So first with neurons, and there's a whole set of figures with neurons. And I tell you, I think in every case I tell you what the markers are for. This is where neurons, we shouldn't have any neural progenitor cells. Neural progenitor cells are the precursors to neurons. They need to make sure that in their differentiated iPSCs that they think are supposed to all be neurons. They wanted to find out, well, is there a small number of the precursor cells to those neurons? Because that would greatly influence their analysis. Okay. Also, they want to make sure that the ApoE genotype doesn't affect differentiation of neurons. Just because they look like neurons morphologically doesn't mean they're functioning as neurons. Right? So that's what they did with neurons. They did that with astrocytes. They did that with microglia. Then they looked at expression. But now they're taking out, when they look at their data, these heat maps, these are true heat maps for expression. Look at this scale. We've got some genes. Are the red genes here being upregulated in the E4 or downregulated in E4 versus E3? These are the ones being upregulated in E4. These are being downregulated in ApoE4 relative to that. But then using these gene ontology terms, go back to some of the January papers you look at. They can go into databases and find out what are the general categories of function for each of these genes that are either upregulated or downregulated. Now, again, this is not definitively telling them that every one of these genes is involved in neurogenesis, or every one of them is involved in cell development, but that pattern is very significant. Okay. Then this is what they could base additional experiments on. Maybe there's a particular gene in here that they're particularly really interested in, and then they could go then and do in the next round of papers, do specific experiments on that. Okay. I had these are plotting the z-scores just to remind you what that was. I'd actually had a slide in the previous paper from January referring to z-scores. Now, so they're looking at neurons. Neurons make synapses in the nervous system of their neurons. If it were neuromuscular, neurons would make synapses with muscles. Let me go forward a slide. So just in case you don't know exactly what neurons do, this is a presynaptic. That's the neuron and sending signals to the postsynaptic neuron, and then these signals would travel. There would be a synapse. This would be considered then the presynaptic to another neuron here, so that's how the signals transmit. And then this is a blow up of that actual synaptic cleft, that there's a little tiny space between the synapse, the presynapse, and the postsynapse, but that's where those neurotransmitters get released from that electrical stimulation that occurred in the presynaptic. There are receptors on the postsynaptic cell that receive those signals, and that's what causes this to transmit those signals further down. That's the essential function of neurons, right? And this takes place in all the different types of neurons in the body, but particularly in the brain. And you've got impaired synaptic function. Remember some of the symptoms of early Alzheimer's: cognitive function problems with the neurons, specifically in the hippocampus. Now, this particular paper didn't differentiate these neurons to a specific type of neuron, but there are techniques for doing that. Other labs have established that, and I wouldn't be surprised if this group or some other group is taking the same approach and then differentiating iPSCs to hippocampal specific neurons versus other parts. That would be very interesting. So then, I think I told you previously, I don't understand these electrical stimulation pathways, but you can see, let's look at just their data. There is a difference, okay? Oh, and on every one of the slides, if you don't know this convention with asterisk indicating the p-value, I've added them. So basically a p-value of 0.05 would be less than 0.05 is 1 asterisk, less than 0.01 is 2 asterisks, less than 0.001 is 3 asterisks. And they do have some of their values. These are actually pretty tight values, but some of them really aren't all over the place. So if you weren't, and what another thing I like about this study, not only were they revealing from this analysis that there was altered synaptic function with respect to the APOE genotype, they're following it up. Well, if this is true, if this is real, they should see a difference in the quantities of these two markers. One of them is pre, the other one is post. So it's a nice way of looking at, are both of those genes, these proteins, are this their cells, the proteins in these cells, are they being expressed? And yes, they are. But they are not consistent with different synaptic function. There's a difference with APOE4 versus APOE3. Now, still a long way towards understanding how this contributes to Alzheimer's pathogenesis, but it's one little piece of information that we follow further. Okay, endosomes have to do with endocytic pathways, so what neurons do is they take up the EEA1 is a marker specifically showing them where the endosomes are. So, and this is relating to the fact that these neurons, we're still neurons, that the secreted levels of these A-beta peptides, remember the processing, right? So only the pathogenic pathway is producing higher levels of A-beta 40 and 42. They're also increased, depending on how you normalize it. So this is increased in APOE4, that's increased too. So there's more secretion of A-beta, and there's also a lot more early endosomes. So what they're doing is taking back that A-beta and basically recycling it inside the cell and presenting it back for endocytosis. And this is something these increased numbers of early endosomes have been observed in other both tissue culture models and in actual brain samples with Alzheimer's, but it wasn't clear what the significance was. But how this relates directly to that, they don't show here, but it's clearly implying that these neurons with APOE4 genotype behave very differently with respect to both exporting A-beta and taking it up, recycling it. Okay, now we're getting into astrocytes. So that was a whole series of analyses, right? Descriptive studies, really, about neurons. Now a series of descriptive studies with astrocytes. Okay. Again, I tell you the markers. Okay. Now this is really pretty cool. This is showing you expression of APOE is where it's supposed to be, but look, it's in astrocytes. That's where the vast majority of APOE is expressed from. But APOE3 genotype has a lot more expression, at least at the level of the protein, than do the ApoE4 phenotype. I'm not sure there was any reason to suspect that before. Okay. And they quantified it here. Now that doesn't, there's only a p-value of less than 0.05, but look at this range. So that's a problem with what they're doing here is using fluorescence, digitizing techniques with the fluorescence to try to quantify this number, and that's why there's so much of a range. I think it's not a real good quantitative assessment. So take that with a grain of salt. But they followed that up. They didn't, expect us. I'm going to go back a slide. They didn't expect us to take their word just on these data, right? Especially with that big range. They followed that up, looking at the level of actual protein using Western blots. Okay. Now, pay attention again. This is information that you would get out of the figure legends, but I saved you some time. I put them for cell pellets, right? So that's removing the cells from the culture dish and the media. Okay, right? Cells are grown in culture. They're attached to the bottom of the dish. Anything they secrete from the cells is going to be present in the media that you have, that liquid that's on top is feeding the cells. And that's what conditioned media is. This wouldn't be media right after they've added fresh media to feed the cells. This is media, I don't think they said, but 48 hours or some time period after they've added fresh media to the cells. Any protein that would have been secreted outside of the cells would be present in the media. So combining what's present in the media along with what's present in the cell pellet, so that's spinning down, centrifuging down the cells, and then lysing, doing the Western blot on the cell lysates. Okay. So again, there's a big difference between the amount that's present in the cells and the amount in the conditioned media. Yes. Oh, yeah, that's a good question. I didn't actually look it up, but that's something that you do. That would be a post-translational modification, adding sialic acid. Anybody work in one of the carbohydrate-intensive labs here, like adding a carbohydrate post-translationally to a protein? I'm not sure what that's doing, but it is adding a sialic acid. It could be not having anything to do with the function of it. It might be directly affecting its function. It might indicate some differences in the processing of it. I don't know. Any of you biochemistry people know what sialic acid modification does to a protein? You're a biochemistry major. What's sialic acid? Like I said, he's a biologist. Yeah, you were being pointed out. Yeah. Somebody look it up and tell us.
I feel like it's different based on different proteins.
Different what?
I feel like it would be different between different proteins.
Yeah, but so remember these differ with only actually one amino acid difference. So I would say either ApoE4 has an amino acid where it gets sialated that ApoE3 doesn't have. And whether that has anything, I'd be interested to know if ApoE2, now I can't remember, in one of the review articles you can see what the amino acids, I think I might have on a slide from Thursday telling you the amino acids that are different between ApoE2, ApoE3, and ApoE4. E3 and E4 differ with only one. Anyway. Okay, and then again, gene expression. Now let's look at these. Let's think about this. With both the astrocytes and neurons, the ApoE4 cells have a very pronounced different transcriptional profile than the ApoE3. Does that say that ApoE4 is a transcriptional regulator, or ApoE itself is a transcriptional regulator? ApoE what is ApoE? Is it could it possibly, based on what you know of ApoE, could it be a direct transcriptional regulator? Meaning, is it the ApoE itself directly affecting expression of all of these genes? Nobody wants to hazard a guess, an educated guess, a reasonable, informed. You look like you're thinking. I'm picking on you, Nikita, right? Nikita?
Well, it could be just the influence of the immune response to ApoE4.
This gives us absolutely no evidence that it's doing it directly. In fact, everything that we know about ApoE, right, it's being secreted in the cell. It carries cholesterol around outside of the cell. Now, that doesn't mean it could, it'd be impossible for it to be doing directly transcribing, regulating transcription of some genes, but it's pretty unlikely. These are downstream effects. That's what I was getting at. The different ApoE genotypes are influencing a lot of other things inside the cell. That's what's causing these profound differences in gene expression. But again, you look at the gene ontology groupings, and they're significant to what you could hypothesize might be relating to how the ApoE genotypes contribute to pathogenesis of Alzheimer's. More about astrocytes. Everybody clear with FACS analysis? With the Collinson et al. Paper from January set up. This is cell sorting, okay, and they're staining. I'd never heard of this Philippin 3, but when it binds, it's well characterized. I saw a number of, Sigma and a bunch of other companies that sell it. But it's not fluorescent until it binds to cholesterol. So they just add it to the cells. Any of the cells that have cholesterol are going to have fluorescence. And then they can quantify it again. And one more thing, this is just really nice, I think just really good. It's descriptive, but I think there's great value in descriptive studies because they're laying the foundation of the biological properties of the system and establishing that they're relevant. Okay, so this is following up these observations with an independent method of quantifying. So this is just staining on a per cell basis. How many cells have a higher level of fluorescence, meaning higher level of cholesterol, and the results are exactly reproducible. They have less uptake. Now this is using, don't worry about what an ELISA is. Have you heard of ELISA, enzyme-linked immunosorbent assays? It's an antibody based with an enzymatic activity as a readout. Okay, but it's just a really nice sensitive method for quantifying where you don't have to do a Western. Okay, so this is just, they can add a beta-42, the protein, it's a small peptide. I can't remember if they where they purified it from, but they purified it and then again just added it to the media. And 2 days later, they just harvest the cells and then get the conditioned media, quantify the amount that's present in the conditioned media, and then just subtract it from the amount they put in. Or based on the total number of cells. Based on the total number of cells is normalizing if there's some small differences between the number of cells in each dish, but reduced. And validating intracellular, now we're at the microglia. Validating. Now this is what they were careful, and we're looking at the morphology of the cells. They were very focused on doing the protein evaluation. Were these cells expressing the cells they should be? Are they expressing the right genes? But they were also careful to notice the morphology. And does anybody remember what the major functions of microglia are? What are they related to in terms of function? on other cells that aren't in the brain. These are specific for brain. Do they have synaptic function like neurons? They're like an immune, they're like macrophages. They basically clear away debris in the brain, right? And we don't like to think about debris in the brain. I think I've done a lot of debris in the brain today. There's more some things than they're technically. I've got bad functioning microglia anyway. But they notice these processes, so those processes extend out. Now I'm not sure how if these look exactly like they would inside a brain, but clearly they know that these processes extend out. They see difference of morphology, so they quantified them. And they wanted to make sure they didn't have any contaminating neurons or astrocytes. Those cells send out lots of processes, right? They wanted to make sure that the cells they were looking at were only the microglia cells, not some contaminating neurons or astrocytes. Same expression, gene ontology. But look at this. Immune reactive genes are being upregulated. This is an aha moment, I would say, because they know that inflammation and basically overreaction to the early aspects of the pathogenesis of Alzheimer's is key to the further progression of the disease. So here we've got these macrophages with the ApoE4 genotype that look like they're being hyper-responsive for a lot of the immune functions. And by the way, this reminds me, if you don't remember, or you might be able to figure it out from here, which is the genotype, ApoE4 or ApoE3, the risk factor for Alzheimer's? Is it 4 or 3?
4.
4, yes. So this clearly could be relating to that risk susceptibility. And this is really cool. You can go online and get the video microscopy, but these are just stills. So this is taking up A beta 42. ApoE3 cells just grab onto A beta 42 and just suck it up. Remember, that's clearing out all that debris. You don't want A beta 42 accumulating. That's contributing to the plaque formation. ApoE4 microglia are defective for that. So not only are they defective for the immune process, they're upregulating their immune processes, but they're defective for taking it out. Now this is where they get into potential cell-to-cell interactions going on. This is where they use organoids. And I wish we could spend more time talking about these iPSC-derived organoids because they're just an amazing advancement, I think. So they can let these cells grow in three-dimensional culture. Instead of just having them attached to the dish. Yeah, you can have two different types of cells attaching to the dish, and you can get some estimate of the interaction between the cells, but you're not, with a 2D culture, you're not recapitulating the three-dimensional interactions that can take place, which, again, have to happen inside the tissue. So what they did was APP-DP, so DP is a duplication. So this is one of the pathogenic mutations. What's APP? What's the significance of APP for Alzheimer's?
It's a precursor of the A-beta protein.
Amyloid precursor protein. So you could have mutations in APP that disrupt the cleavage sites for the secretases. Another one of the mutations is a duplication of APP. But what they did was they were able to make these organoids from Alzheimer's, that have the Alzheimer's genotype, but then introduce into these organoids, so microglia can move around like other macrophages and see if they can penetrate into the organoid, and then what happens. And this is where they're getting the three-dimensional evidence for three-dimensional differences. What's the significance of, let's see, okay, A beta? IDA1 is the marker for microglia. That's being upregulated. These cells are being upregulated when they're getting activated. So there's something about, these are the ApoE3, these are the ApoE4. This is telling us that they're functioning normally, but then now look at the staining for A beta. ApoE3 doesn't have the accumulation of A beta. ApoE4 does. Okay, so ApoE3, remember, without the microglia, these organoids, the Alzheimer's organoids, are making these aggregates of A beta. If this were left alone in this door in the brain, these would develop into the plaques. A beta E3 phenotype, probably because of the differences in microglia function that we saw in the previous slides, don't have any of that accumulation of A beta aggregates. E4 does. Now E4 is clearing them out, so it's not like it's completely defective for function, but a clear difference between E4 and E3. Okay. What's p-tau? Why are they concerned about p-tau? That makes the tangles. Tau is an essential protein in these cells, but for reasons that aren't completely clear yet, it gets hyperphosphorylated during the course of Alzheimer's disease. And that's what causes the tangles. This is, again, if you go and look online, you can see the 3D reconstructions of these guys kind of turning around. It's really cool. So remember, these are the questions you should be thinking about with every one of the papers. We're going to get together and talk amongst yourself, outside of class, talk about them, discuss them, come up with some good ideas. I'm not expecting, huge technical expertise with proposing experimental approaches, but, something like tissue culture, iPSC-derived, Western months, microarray, RNA-seq, that kind of thing. Okay, let's take a break.